it's my honor to introduce Dr. Stephen Baenziger, who is the Nebraska Wheat Growers Presidential Chair in the Department of Agronomy and Horticulture. Dr. Baenziger obtained his bachelor degree in biochemical sciences from Harvard University in 1972 and master and PhD degree from Purdue University in 1975. He is the primary small grains breeder at the University of Nebraska, where the major goals of their programs are cultivar and germplasm development. So far, he has co-released 33 wheat, 5 barley, and 4 triticale cultivars, and over 20 germplasm lines or populations. So let's welcome Dr. Baenziger.
Well, first of all, thank you, and can everyone hear me? Won't ask if you all want to, but at least you can. That's the good side. Okay. As mentioned, the title of my talk is The Germplasm and Its History of Common Wheat. And I, first of all, want to thank the organizers very much for inviting me. It's always great to have graduate students invite you, and I truly appreciate that, and it's wonderful to come to Iowa State University. Would like to acknowledge my co-authors, Marta da Silva Lopes, Ibrahim Salah, Ayamani Avachan, and Mary Gutierrez. They all help in the landrace talk of this presentation we're going to talk about. Now, what I wanted to do is I wanted to basically give you the topics we're going to do. The first one we're going to start with is where wheat's grown today. So we'll get you a snapshot of the present. We're going to talk about the origin of cultivated wheat because we want to let you know how you got to today from where you've been, how we understand our wheat germplasm, how we source our wheat germplasm, how we understand the environments. Then we're going to come back about 150 years or so and look at how wheat came to the Great Plains or how it changed. There was a major event that occurred. And then we're going to talk a little bit about how technology is changing. And it's great to follow Dr. Miller because he did a beautiful job leading into many of the topics we're going to discuss. So, first of all, how many of you work on wheat or have ever experienced wheat? Just got to ask. This is a corn and soybean state, okay? So how many we got? We got a few. Okay, that's what I wanted to see. Okay. So I've got to set the mood, okay? We've got to get you thinking like wheat people. So this is where we start. Socrates, the cradle of Western civilization, the cradle of democracy, okay? What is this comment? 2,300 years ago. And I apologize for the gender specificity, but we'll try to make up for that, okay? No man qualifies as a statesman who is entirely ignorant of the problems of wheat. Now that has two aspects to it that I want to bring to you. Most of you may be unaware, but the Arab Spring coincided with a spike in wheat prices. And North Africa and West Asia are where the highest consumption of wheat is. So wheat is immediately important to a very volatile area, okay? That's the first thing. The second thing is Iowa has a reputation for being sort of oversized when it comes to picking candidates and whatnot for elections. And I'm wondering if you guys are doing your job and asking the real questions, okay? I mean, when you think about Washington, are you asking the right question of the candidates that come through here? Just think about it, that's all. So where was wheat grown? Now, first of all, and I apologize because the colors are probably hard to see, but there are major classes of wheat, okay? And they each have different uses. So in the central Great Plains, the green dots are hard red winter wheat. You'll see a smattering of purple dots. That's hard white winter wheat. That's all bread wheats, used predominantly to make bread. If you go to the east, you'll be the soft wheats. That's the cookie, the cakes, the crackers, soup thickeners, wallpaper, paste wheats. If you go into Michigan and parts of Wisconsin and the PNW, that's soft white wheats. That's your cereal wheats. That's also ice cream cones. And then in North Dakota, you'll see the durum wheats. That's the red. And also the desert durums, which are in Arizona. And then the golden wheats are the hard red spring. They're mainly used as bread wheats also. Okay? Durums are your pasta wheats. Okay? So if you eat good pasta, that's what you eat. That's where wheat is grown today. Now, if you want to think about the origin of your germplasm, you have to remember a couple of things. That history celebrates the battlefields whereupon we meet our death, but scorns to speak of the plowed fields whereby we thrive. It knows the names of the king's bastard, but cannot tell us the origin of wheat. That is the way of human folly. That's actually true, okay? Because if you look at the origin of wheat, and what we want to emphasize in this slide is wheat is a polyploid with immense genetic resources, okay? 60 million years ago, the Poidiae evolved. 35 million years ago, the progenitor of wheat most likely evolved, totally lost in history. And from the progenitor, we went through what's called divergent evolution. Okay, the progenitor had 7 chromosome pairs. All of the first line we can see here, which would be, and I'm going to see if I can do this. Okay, can you see the little cursor up at the B genome? Whoops, went the wrong way. That's not what I meant to do. Okay, see the B genome? We don't know what that is. We presume that it may have been a hybrid of, or a progenitor of what we think are the current B genomes, but it's unknown. 1 million years ago, the A genome evolved. That's the Triticum uarta. Okay. The C genome, which we're only going to mention for a different reason. The D genome, which is the Triticum tauschii agelopsclerosa, depending on your nomenclature, 2.5 to 4.5 million years ago. Barley, which is derived from the exact same progenitor, 11 million years ago. Rye, 7 million years ago. That's the divergent evolution. Of these, we have a very good idea of what the A genome is, a very good idea of what the D genome is. Of course, we know what barley and rye are. We don't know the B. About 500,000 years ago, there was a spontaneous hybrid. This is convergent evolution that formed the Triticum dictycoides, which are the progenitors of the durum or pasta wheats. Okay? Now, I put that up because you can have convergent evolution in non-crop species also. So the C and the D genome also hybridized, and they formed jointed goat grass, which is one of the nastiest weeds we have in wheat. So it's not like evolution only gives us great food crops. It gives us weeds too. What's interesting is about 8 to 10,000 years ago, the dictycoides spontaneously hybridized with the tauschii to give us common or bread wheat. Okay? And about 140 years ago, the dictycoides, or durum wheat probably, hybridized with rye to give us triticale. So the idea of convergent evolution is not something which is ancient. Now, what do you need to think about this? Okay, I mean, what's the important part about this? If you're looking for genetic bottlenecks, there's 500,000 years of evolution in the dictycoides. There's 8 to 10,000 years of evolution in bread wheat. The D genome is where the least variability is. Reason being, there were few spontaneous hybrids, and there were relatively few years for that to evolve, mutate, and do all of the other things, okay? Now, how do we know this works? It's because we can retrace the history. Now, we don't know the B genome. We put up Triticum speltoides, which is here. Why is the B genome, why is wheat always ABD, but in fact the B genome? It's because the cytoplasm is from the B genome. It's not from the A. We know that. We know it's not from the D. So the B genome must have been the original female. Okay? The A genome, this is representative. It's Triticum uarta. This actually is Triticum monococcum relative. That forms the Triticum dictycoides. Okay? And quite easily you can hybridize dictycoides with tauschii to get bread wheat. So we make what are called synthetic wheats. We're recreating it, and it's done routinely. Where did this occur? Well, it's the Fertile Crescent, right? And when you think about domestication, you think about where the polyploidization had to occur, there's 2 key points. You have to have where the species overlap, right? They have to be close enough they can hybridize. That happened in the Middle East, okay? And that's where the polyploidation, that's where the greatest treasure trove of the wild relatives of wheat are. Okay? Now, if you've never seen what wild wheat looks like in the field, this is a picture. This is the Golan Heights. This is occupied territories, currently in Israel, formerly in Syria. What I'm showing you is Triticum dictycoides, okay? There would be more plants of Triticum dictycoides in a field like this than there is in a commercial field of bread wheat or durum. That may sound strange, but if you think about it, all the seed shatters, so the seeding rate is immense. Even if the yield is only 1 or 2 bushels per acre, the seeding rate is astounding. Okay? Now, wild wheat and wild oats, which you can see here, coexist, as does wild barley. The difference is that wild barley has already shattered out and you can't see it anymore. Okay? But this is what a native stand of the origins of wheat would look like. And just to put a little perspective here, if you're ever in Syria, this is a Crusader castle, the Croc de Chevaliers. It was captured by Richard the Lionhearted. He built a new entrance on it, and he put 2 lions up there so everybody knew Richard the Lionhearted had conquered it. Okay. It's a massive fort. And the interesting part is, if any of you know the poem Ozymandias by Shelley? Anybody ever heard of that? Okay, I see one in the back. Ozymandias is where an ancient king boasts that Ozymandias, the king of kings, all you that see this bow down in fear because I've been so great. Nothing you can ever do to match. And I put this in because where the baths were is where the wild barley has reclaimed its native home. Completely deserted, lost castle, and the wild species come back and they reclaim their home. Okay? So if you ever have a chance, Ozymandias is a good poem. It's short, you can read it very quickly. Now, how do we classify wheat germplasm? We classify it mainly by adapted cultivars and experimental lines. We have then unadapted cultivars. Wheat is the most widely grown crop in the world by acres and probably by ecological diversity. We have winter wheats. You know, you think about how cold it is when you came to work here. The wheat's sitting out in Nebraska just saying, hey, we can take this, no problem, okay? We have spring wheats, sort of like the sissy crops that, you know, soybeans and corn are. Sorry, guys. Just have to tease and the like. But we do have some things that do that. We have land races, okay, which we'll talk about quite a bit. We have the wild relatives, the A, B, and the D genome donors, but we also bring in genes from a lot of other wild species because we all have the cytogenetic tools. The type of questions we have to ask when you think about this, in evolution there are rare events in the polyploidization. Did we get the right A? Did we get the right B? Did we get the right dictycoides? Because the dictycoides itself became a bottleneck when you crossed it with the D genome. Okay? That's where we get our synthetic wheats. And if you look at triticale, where you swapped out the D genome for the R genome, is the D genome even the whole right genome to be using? Okay? So this is how we begin to classify our germplasm. In a breeding program, 95% or more of your crosses will be elite by elite. I don't want to have you think anything other than that. At the same time, what I want you to understand is how the germplasm works and how it flows. So while I'm going to talk to you about things which we don't spend as much time on, they become very important foundations. But never assume that an applied plant breeder is making, never assume that their majority of their crosses is anything other than elite by elite. We do elite by unadapted. We'll talk about what that means. We do elite by land races. I'm going to give you some pretty good examples of why we do that. And then we go back to the synthetic wheats and wild relatives, basically to broaden our gene pool. And if I told you today that the elite nurseries coming out of CIMMYT, the International Maize and Wheat Improvement Program, 25% of their best wheats all have synthetic germplasm in them. That shows you how valuable the wild relatives are, how valuable the bridge from the wild relatives to modern wheat are, and that why evolution leads to successes, it doesn't always give you the very best. Okay? Evolution is random chance. It just has to be better. It doesn't have to be best. Okay? So again, most of these are from elite by elite crosses. When we use the unadapted germplasm, we usually do it through a back cross. The conversion program is a perfect example of why we do that. The elite or the Landraces are used for where we don't have traits in our elite gene pool, and this is a very active area. Okay, synthetic wheats are the bridge. I'm going to end with a few bits about mutations in cytogenetic or cytoplasms, and I think the sterile cytoplasms in sorghum are a great example. This is last year's preliminary trial. Okay. We do a lot of genomic selection. One of the things you can do is you can look at it for diversity. We're always trying to make sure that we are keeping our diversity. If you look at the bottom, which is in red, you can see that's the sort of the main lineage of the Nebraska gene pool. 3 good cultivars, Camelot, Goodstreak, and Settler CL are there. The most popular wheat in Nebraska up to last year was Overland. Okay, it's got its lineages now beginning to come into the program. The second to last release we've just made is Freeman, a very good nitrogen use efficiency. It's coming out of a different lineage, okay? Has a lot of alliance in it. The green and the gold lineages, no cultivars are in those lineages. Interestingly, as we advanced line, the gold lineage disappeared. We went from 270 lines to 57, and the gold lineage disappeared. So I'm thinking I probably should have gone back and checked that a little more carefully. Okay, if I look at my own breeding program, most of the lines are from the elite by elite crosses. The clusters I've just shown you do fit the ancestral lineages. Okay? Many of the unadapted germplasm, they're really parent-building lines. You use them, you cross them, you suck them back in. They're sort of a slow-moving recurrent selection program. Okay? In my program, we have relatively few lines with Landraces, synthetic wheats, or unadapted types of the wild relatives, okay? Other than for genes for resistance. Now, how do we use our germplasm? And this is something where the wheat community, I think, still is what I would call a community. We have great regional trials. So what this is the Northern Regional Trial. Every university and private company in the Great Plains that wants to see how its lines do in the north, they submit entries, okay? We have 30 years of data on this. Jim Peterson, formerly of the USDA and at Lincoln, was the coordinator, and he said, how do we cluster based on yield? And what you can see, if you go to Nebraska, for those who are unfamiliar, that's this little state here, okay? The eastern sites cluster with the eastern sites in the Great Plains. The western sites cluster with the western sites in the Great Plains, mainly Wyoming. Now, he did the same thing for the Southern Regional Performance Nursery, a lot more testing sites, okay? And so when he did that, you begin to see again now Nebraska actually has 3 clusters. It has the eastern site, which is the eastern Great Plains, the western sites, which are the western Great Plains, that'd be Wyoming, eastern Colorado, northwest Kansas, and then Alliance, which is the star, actually correlates with South Dakota and going into Montana. Now, if you think about that, why are these nurseries important? First of all, they're a great place for germplasm exchange. Secondly, it gives you additional testing sites that are relevant to your germplasm. So if you lose something to winter killing or hail or drought or any of the other maladies that happen in wheat, you can go look at eastern Colorado, northwest Kansas. You can look at eastern Kansas, depending upon what you're missing. You have a great way of knowing how broadly adapted your lines are. Okay? Lines from similar regions have similar adaptation genes. So if you're looking at your program for elite germplasm, you go to Colorado and you say, I want something for my west. I go to eastern Colorado, I go to northwest Kansas. That's what I'm looking for. Now, a tip. As a plant breeder, every breeding nursery will tell you a story. Your data, when you get it, if you don't know the story behind it, it's like dissecting a corpse. What you really want to do is to know the story that told you how to get your data. Okay? And this is why these regions become important. Now, How do I actually use that data? If you look at the blue dot, that's Lincoln. The gold dot is North Platte. The red dot is Alliance. If I have a line that tops those three nurseries, I have a line that will do well in the eastern Great Plains, the central Great Plains, and the northern Great Plains. I own the northern Great Plains. So when we released Overland, it topped the regional trial, topped all across Nebraska and everywhere north. Okay? Freeman did the same thing. It tells me how to put my data together. It tells me what I want to make crosses with, and I can figure out what I'm doing. Now, the importance of germplasm and the history. Often elite lines do not have all the traits that we want. Every gene pool is limited. That's why you go to the conversion program. That's why you go to tropical crops to find things for your temperate regions. How do we sample the germplasm that's not in the Great Plains? Well, you try to stay as close as you can until you find out that gene pool is missing what you need. The unadapted cultivars, the Landraces from similar regions of interest that may have the trait, and then finally to the wild relatives and synthetic wheats. So Jim Peterson, in addition to coordinating the great regional nurseries of the Great Plains, he coordinated the International Wheat Performance Nursery, which was grown globally. And so he said, let's take a look at the clusters there. So in the first cluster, now this cluster is not quite as perfect, because again, remember, your clusters are only telling you what you're close to. You know, if you change your inference space, things that were close before become distant and vice versa. But if you look at Lincoln, Nebraska, with 14 years of data involved in Region 1, there were 7 regions, Ankara, Turkey clusters. Okay? You can see a lot of the Great Plains, things like Akron, Colorado cluster. Okay? Ithaca, New York, only because it's closer than the other 7 regions, other 6 regions were. If you go to Region 2, you see what we would call the PNW, the Pacific Northwest, which is Pullman, Washington, which fits very well with the great European wheat belt that extends from Paris all the way to Moscow, places like Wageningen and whatnot. Okay? That's the clusters. Now, the reason why we bring this up is if you went back 140 years, okay, almost 150 years ago, Nebraska was a spring wheat state. Northern Kansas was spring wheat. Now, if you look at this, you can see winter wheat now extends all the way into Montana. Okay? What changed in 1870? They brought in one wheat, Turkey Red. Okay? Now, I'm going to let you read this, but the brief synopsis is the following. There were Mennonite farmers who, due to universal conscription in Germany, fled Germany to Russia. Okay? Catherine the Great said, we need farmers, we got lots of soldiers, you can be farmers, we won't put you in our army. They came. They were very prosperous farmers. Catherine the Great died. Later on, a few czars said, we need a lot more soldiers, universal conscriptions. The Mennonites are pacifists. They emigrated from basically the Crimea region, places where Russia and Turkey fought over for years, to the Great Plains. With them, they brought their seed. Okay? So that's the change. This is a historical marker in Walton, Kansas. Okay? Mark Carleton, who was the first president of the American Society of Agronomy, was a great plant explorer. He didn't discover Turkey Red, but he popularized it because he was always looking for a wheat that would survive the winters of the Great Plains. Okay? It needed to survive harsh winters and hot, dry summers, which are what's known as the steppe wheats of Russia and Turkey. Okay? Ankara of the Anatolian Plateau is part of that steppe region. It does cluster with Lincoln, Nebraska. Okay? For wheat, we get a statue. Okay? So that's a statue of the Mennonite farmers in Newton, Kansas, that actually brought the wheat in with them. And if, for those of you, and I don't know if Kendall's still here, but your old plant breeding textbooks, this picture is actually a current one. That's my grad students. I always take my grad students by there every time we go by Newton. But in Briggs and Knowles, there's actually a picture of this statue. Now, Every talk you have to learn a new term, right? Okay? So what is a landrace? It's an autochthonous wheat landrace, is defined as a traditional variety with a high capacity to tolerate biotic and abiotic stresses, resulting in high yield stability and an intermediate yield level under low input agriculture. Anybody know what the word autochthonous means? No kidding. Okay. Okay, great. This is good. I had to look it up. Now, tell me if I'm wrong. It means indigenous, and it means, the definition I like is formed in the place it's found. So a landrace is something that's grown in place, and it's indigenous to that, and it's formed by where it's grown. Okay? A landrace differs from a variety that has been selectively improved by breeders to include particular characteristics. That's a little bit of a misnomer. Landraces are often bred very well by very good farmers who are the original breeders. But that's how they're defining it. Now, I'm going to give you a diversion, but I want to come back, and hopefully this will give you a little idea of germplasm use and how we figure things out. Okay? So when we wanted to study grain yield, we started in the 1980s. Okay? There were extremely few molecular markers in wheat. Most of them were between wild species and cultivated types. Within the cultivated gene pool, there were virtually none. Okay? We had incredible cytogenetic stocks, and Rosalind Morris devoted her life to recreating reciprocal chromosome substitution lines. I don't know how good you're being taught in cytogenetics today, but this is a really interesting contribution. And a couple of things I should say is, do you know that March 2nd was the International Day for Women? Anybody know that? Okay, kind of thought it passed kind of unsaid. CIMMYT put out the Super Women of Maize and Wheat, okay, that made a big difference. And if you Google Super Women of Maize and Wheat, you will see Rosalind Morris is listed as one, okay? And it's a really great story about her. She's one of my heroes, actually, okay? But she took her life, and she said, we have Cheyenne, which is the foundings of the Nebraska wheat breeding program, and we will replace chromosome by chromosome, each chromosome of Cheyenne, 21 chromosomes, pairs, okay? We'll replace each pair with one out of Wichita, a very popular cultivar out of Kansas, okay? And we'll do it in reverse. We'll take each chromosome in Wichita and we'll put it in a Cheyenne chromosome so we can see what each chromosome did. So when you don't have molecular markers and you're interested in understanding the genome, you have to work with the tools that you have, okay? Now, Cheyenne was the founder of the Nebraska wheat breeding program. When I came to Nebraska in 1986, its coefficient of parentage or co-ancestry is 0.16 with Brule, the most popular cultivar, and 0.43 with Siouxland, which was probably, at the time, the most incredible wheat we've seen. In Kansas, at one of their sites, it went 8 metric tons per hectare, and the nearest line to it went 5 metric tons per hectare. You're working in a state that has an average of 2 metric tons per hectare, and this line beats everything in the trial by 3 metric tons per hectare. It's a pretty amazing line, okay? So this is what you see. Now, I'm going to try to explain this very briefly. On the bottom, the x-axis are the chromosome pairs of wheat. 3 genomes, the A, the B, and the D. They're all homeologous. You have 1 through 7, 1 through 7, 1 through 7. On the y-axis, the 0 point is Cheyenne's yield level. And then when you take the one Cheyenne chromosome out, replace it with a Wichita chromosome, what does it do to yield? Okay? And what you'll see is chromosome 6A from Wichita greatly improves Cheyenne's yield. 6A from Wichita greatly improves Cheyenne's yield. 3B from Wichita greatly decreases Cheyenne's yield. Okay? Like I said, she did this in pairs. This is Wichita. Same slide. By the way, the first slide is 1,000 plots of yield data. The second slide is another 1,000 plots of yield data, just to give you an idea of what it takes to get this. But we were curious. Yield's an important trait. 3A from Cheyenne when put into Wichita devastates yield. 6A when put into Cheyenne devastates yield. And 3B has no effect. Okay. This is the paired slides. We did a lot of work with 3A, and we'll come back to that. So the Wichita chromosome in Cheyenne increases yield by 15%. The Cheyenne chromosome in Wichita decreases it by about 16, 17%. 6A, Cheyenne put in the Wichita chromosome into Cheyenne, 12, 13%. The Cheyenne chromosome in Wichita, 18% roughly. Okay? 3B, when you take the Wichita chromosome and put it into Cheyenne, massive yield reductions, but no effect when you were flipping. How did that occur? 3B has a major winter hardiness gene on it for Cheyenne. As soon as you take it away from Cheyenne, the crop winter kills. Okay? Wichita's winter hardiness is elsewhere in the genome, so therefore when you put Cheyenne's winter hardiness gene in, it adds nothing to what Wichita already has. That's the unilateral side as opposed to the reciprocal effect. Okay? Now we did all the components of yield and things, and what was interesting for this, just for your sake, is that if you look at the Wichita chromosomes in Cheyenne, and I've highlighted them in red, 3A and 6A increase yield. How do they do it? They do it by kernel weight, okay? And a little bit, mainly it's by kernel weight, okay? If you look at Cheyenne and you put it in Wichita, and you then see how did it decrease yield, it's mainly by tillering. So you're changing the phenology of the plant is really what it comes down to, okay? For those of you that remember QTLs, this is a slide where we looked at, so we finally got markers. We were able to break the chromosome apart. And what you can see is there's a very sharp spike, this blue, at one region in the chromosome. There is a second spike that's here, but only when you have all the combined data. As soon as you try to do it by location, you don't have enough power. If you look at that spike, the main one, you can see these are now individual locations. There are 3 environments where you clearly have the QTL. It's significant. The fourth one would not be called significant. This is one reason why because it's underneath the threshold level. It's one reason why I really like genomic selection as opposed to mapping QTLs, because you don't have to worry about are you above or below the threshold. And then you have 2 more regions down here where the QTL has absolutely no effect. That's G by E or QTL by E. Okay? Now, this is a breeder's dream. This is an Eberhardt-Russell plot. Okay? The purple squares are the Wichita allele, okay? Or the Wichita, well, it's the Wichita allele in our recombinant chromosome substitution lines. Everywhere on this curve, if you have the Wichita allele, it's greater than if you have the Cheyenne allele. But the interesting part is that the differences are greatest in your higher yielding environments. Okay, so this is basically using an environmental index. If you go further out the X or the Y, the higher the yield. Okay? As you come back, then it becomes lower yields. Okay? So in the high yielding environments, that's where the QTL has its greatest effect. In the low yielding environments, it has less an effect. So you never lose anything, but it becomes relatively less important. Okay? Now, why is that important? Because we wanted to go back and understand how traits move through a germplasm program. So we looked at clusters of historic to modern wheats, and we looked at chromosome 3A. And what you can see is there is a Cheyenne cluster, which keeps the Cheyenne QTL. The one that we've just said is detrimental, or is in no place positive, okay, or beneficial. How did that stay? The wheats in the Cheyenne cluster are from the drought side of Nebraska, where the QTL has small effects. You look at the Wichita allele, and it's present in a lot of wheats. They tend to be our eastern wheats, where the QTL has a major effect, and our irrigated wheats, where the yields are much higher. Okay? And where did that QTL come from? Well, if you see that red arrow, you probably can't read it. That's Turkey. The closest cluster to Wichita And in there we put in the substitution lines, because that's sort of an internal control. So when you have Wichita, you have Wichita or Cheyenne with Wichita 3A, which you would expect that has the 3A QTL from Wichita on those lines. And if you look at Wichita with Cheyenne 6A, which means it has the Wichita 3A, all of those, the nearest line it clusters to is Turkey. So the QTL that was found in Wichita probably was derived from Turkey. Now it says you one other thing. There are a lot of important genes that have occurred, but if they're not in the right spot, you won't find them. Okay? Every gene has to be understood in the context. The QTL on 3A is understood in the context that it's a high-yielding QTL when you have higher yields. When you have lower yields, it has no effect, or we don't have the statistical power to separate it. Now, that led us to think, let's see, Lincoln, Ankara, cluster in the same set of environments, right? The founding population of the Great Plains came out of Crimea. Interesting. The Turkish wheat efforts began shortly after Kemal Ataturk became president. He built the great wheat breeding stations of Turkey, Eskişehir, Konin, Ankara, okay? Cheyenne, our founding population wheat, was released in 1922. You're beginning to see a little similarity here. 1923, Ataturk became president. He started building the Turkish wheat program. 1922, the Nebraska wheat program's first great release. By the way, the last certified seed production of Cheyenne was in 1979. I've never seen a wheat that's lasted 57 years in certification. And it was grown in Montana, not in Nebraska. And to give you an idea of how these global exchanges work, there was an international nursery for stem rust resistance. Nebraska, I always put it in because it's a very important trait for us. And in 1970, the Turkish breeders asked the University of Nebraska, could they release one of our lines, Bolal. And if you look at its pedigree, what is it? It's Cheyenne by Kenya by Montana. Half that line is Cheyenne. The importance of this is Kenya is a stem rust resistant line. The Cheyenne is the one that basically donated most of the adaptation genes. So you have 100 years of breeding, basically. A Turkish program, somewhat independent. The Great Plains programs, somewhat independent. Can you go back to your source to get new genes? You know, it's like a parallel universe, a replicated breeding program. Can you go home and find new material? So then we looked at Turkish lines. We looked at Nebraska lines. The Turkish lines are the ones in black. The Nebraska lines are the ones in red. If you look at the clusters here, what you can see is there are some clusters where the blue and the black mingle. And I think what's interesting is if you look at Turkey, very close to Turkey is Cheyenne, very close to that is Kharkov, and then Sulin, the wheat that I told you about that was so stunning, is very similar to the original wheats coming out of Turkey. You also see another one where you can see Bennett and Nakota with some of the Turkish wheats. Then you see programs where they're specifically adapted to Turkey, specifically adapted to the Great Plains. Now, again, talking about germplasm flows. Are any of you familiar with the Green Revolution? Ever heard that name? Ever hear Norm Borlaug? Okay. The source of the semi-dwarfing genes was a wheat coming out of Japan called Norin 10. What's the pedigree of Norin 10? And again, I want to highlight germplasm exchange. The pedigree is Fultz. That's a soft red winter wheat released in 1871. Crossed to Daruma. That's a Japanese wheat with all the semi-dwarfing genes. What's the last parent? Turkey red. 50% of Norin 10 is the Great Plains Turkey red. And that one crossed to Brever, which is actually a PNW wheat, became the source of the whole Green Revolution. So if you think about how genes flow globally, there's a little bit of the Great Plains in every modern wheat cultivar. Okay? Now, whoops. And so if you've seen a picture of Norm Borlaug, the equivalent done in rice was by Hank Beechel. Okay? The semi-dwarfs make sense in context. Irrigation, high fertility, no lodging. That's what you needed them for. Okay? So you have genes, put them in the right context, they become valuable. Now, do we still have the step exchange program with Nebraska? You bet. Patrozonka is a prominent parent in our history. Beladorskaya is in Arapaho. Mirnovskaya is a parent building line. Kavkaz is the major parent in Siouxland. Okay? We are constantly exchanging with Eastern Europe and with Russia when you can get the germplasm, because we're constantly going back and trying to pull new adaptation genes in. Now we're going to get into the landraces, and this is work that Marta Lopes does, and was done with Ibrahim, who's one of my former students. If you want to look at the germplasm, and this is all done by GBS, you can see all the colors, not going to spend a lot of time, but turkey is the red that runs sort of right up and down the center. So in landraces, we now have a very good understanding of the diversity that we're working with, okay? So that's the kinds of things that we can do. Now, why do we work with landraces? It's because they co-evolve with the host and the parasite, okay, the pathogen. If you've never had a chance to see landraces or the wild species, sometime you should try to do it, okay? When a new pathogen or parasite comes to the U.S., we immediately go back to look for resistance where that pest or pathogen was endemic somewhere else. In my lifetime, when they opened up the St. Lawrence Seaway, they brought in the cereal leaf beetle, devastated the wheat production, actually worse on the barley and oats in Michigan, Illinois, Indiana, that area, okay? Russian wheat aphid came to the Great Plains in about 1985. Wheat stem sawfly is in Montana and is now moving into Nebraska, okay? So you always would go to where it was endemic to find new sources of resistance. We have much better phenotyping capabilities now than we've ever had before, so now we're going to look at other traits than the easy ones. So they're looking at a lot more things like photosynthesis, drought stress, heat stress, winter hardiness, these types of things. And wheat, being the most widely grown crop in the world, has tremendous resources. This is a paper that just came out in Crop Science. It's from Kansas and Oklahoma. And I just wanted to show you how landraces are used. Russian wheat aphid 2 is a new virulent biotype. Very few genes for resistance. They screened cultivars from China, advanced breeding lines, 505 landraces, 31 genetic stocks. The cultivars from China, the elite lines from North America, all susceptible. They found 9 landraces which were uniformly resistant, and they found 28 other landraces which were heterogeneous. Landraces are always heterogeneous, okay, or almost always, okay? So you have to go in and repurify them, and that's what this paper is all about. But think about it. They looked at a number of lines. The only place they found the resistance is in the landraces that came out of the area where the pest is endemic. That's how we use it. So our original source of Russian wheat aphid resistance came out of Iran. Okay? We go back to where the pest came from. Now, coming back to synthetic wheats, because these, again, are extremely important. And the question is, did we get the A, B, and D genomes when we polyploided hybrid wheat? So originally, the synthetic wheats concentrated almost exclusively on the D genome. It was easy. Durum wheats have AB. The diploides already hybridized, okay? Add the D, make a whole set of things. You usually screen the D genome, find out, looking for traits you want, then you make the synthetic wheat. There's less used, but some interesting work now being done on saying, did you get the right A genome? So now you're making diploides crosses by Uarta crosses to try to expand the diversity within the A genome, okay? Can't do anything with the B genome, really. I mean, you can do with pseudo-B genomes, but you don't have the thing. And then again, coming back to triticale, which is a biomass crop, a forage crop, a lot of different things, you may discover that the D genome was by luck, is what led to common wheat, but the R genome or some other genome could actually be better. And for biomass, the R genome certainly is better than the D genome, okay? Now, I'm going to want to spend a few minutes talking a little bit about mutations, because we're talking about variation, and I don't want you to think that all of our germplasm only comes from crosses. Now, there is no transgenic wheat anywhere in the world because of consumer acceptance. I hope to live so long to see transgenic wheat. I'm a big proponent of that. But right now, commercially, there's none. Mutations. There's two types of mutations. One's chromosomal breakage. And so this happens to be, if you can see the yellow, that's 1RS, okay, added to 1AL. So you have on chromosome 1A of wheat, you take off the short arm and you put in the short arm of rye from the homologous chromosome. Put it in perspective, there's more DNA on that one arm than there is in the Arabidopsis genome, and it's totally unregulated, okay? So when you think about genetically modified crops where you put one gene in and you get a lot of trouble, and I put bigger than the Arabidopsis genome in from a different species and nobody cares, don't tell the news, okay? But it just puts it a little bit in perspective, okay? And on the one RS, there was resistance initially to stem rust, leaf rust, stripe rust, powdery mildew, and green bug. And it all co-segregated in one unit because it doesn't cross over with wheat. Kind of a nice thing, okay? Now, a lot of those resistances have broken, okay? Now, the other thing is, I told you about jointed goat grass. We're trying to tie all these little tails and diversions together, okay? Jointed goat grass is susceptible to the imidazoline herbicides, okay? So is common wheat, okay? They share the D genome. If you spray wheat with an imidazoline herbicide, it kills it. So the wild type wheat is the one that says S for susceptible. If you let it grow to maturity, it's dead, just as would the jointed goat grass be. If you have a mutation, it makes it resistant, and you can see what it looks like. That's probably some of the most popular wheats being grown in the Great Plains right now are mutations for herbicide tolerance. Now, the final frontier, if you're thinking about what's going on in the future, is hybrid wheat. And this is the slide that never goes out of date. Hybrid wheat is always around the corner. You could have used it 20 years ago, 10 years ago, this year. I think we're getting closer. Hopefully we're getting better. There actually is being grown in India, South Africa, Europe, and Australia. Okay, seed costs are a major impediment. There's a lot more work and a lot better chemistry on chemical hybridizing agents, and then the CMS. Okay. This is, and I want to use this because, again, the cytoplasmic variation is often overlooked. We think of nuclear variation, but we forget about cytoplasmic variation, and I think that's an area where we should really look a little harder. That's what a true CMS looks like: gaping florets, no anthers extruding. Partial restoration, you get some seed set, but not much. Full restoration, this is all genetically done, is complete. Okay. When you're going to test hybrid wheat, you test at a low seeding rate because the cost of goods is what kills you. Seeding rates for wheat are very high normally, okay? So you have to reduce the cost. This is the program in Mexico in India. Again, their steriles, what their fertiles look like. That's their crossing block. In the upper right corner for you, you can see how they've made the pollinator. Basically it's like 4 feet, then a pollinator, then 4 feet. You can do that if you have inexpensive labor and small equipment, okay? Pretty hard to do that when you've got a 20-foot combine, okay? So this is how they get past the self-pollinated lack of cross-pollination, okay? But you can breed for that. And by the way, there's just a new paper showing that chromosome 4R from rye greatly enhances the pollination in wheat when you add it as an addition line. So we think they could be used for CMS, for the hybrid wheats. But again, just because we got the B genome cytoplasm doesn't mean necessarily it's the best. We should be looking for other cytoplasms. And in fact, there was an old paper by Bob Bush. I don't know, do we have anybody from Minnesota? Okay, the Minnesota table. Do you ever, Bob's now deceased, but you probably heard stories. When he was at North Dakota, he showed that Triticum maca cytoplasm actually had higher yield than the Estivum cytoplasm in alloplasmic, which would be isogenic lines, but for the same cytoplasm, alloplasmic lines. So maybe maca is one we should be looking at. Now, what are our conclusions? Wheat's a global crop with global germplasm exchange. It's a big world. And the germplasm exchange, I think, in wheat is still one of the best. We have our lawyers, but nothing like what other crops seem to have. In breeding, you have to select your parents very carefully, elite by elite, elite by unadapted, elite by landraces, then down to the synthetic wheats, okay? But I would say we've never had better tools to evaluate our germplasm than we have now. We've never had better ways of doing high throughput phenotyping than we have now. And the cool part is they're only going to get better. So the idea of what you can do with markers, what you can do with phenotyping, what you can do with your theory of breeding is only going to be better. Now there's an old expression, proverb comes out of Africa. If you want to go fast, you go alone. If you want to go far, you go together. And so one of the points I want to make about germplasm is the free exchange of germplasm is to a benefit of all. And if your crop has lots of limitations on germplasm, that's a hindrance, okay? And so if you look at the challenges that face germplasm and breeding today, it's best if you can go together. Now I'm going to end on what the Nebraska Small Grains team looks like. We have a lot of people working in the genetic side. We work with plant pathologists, variety testing. We have our own transformation group. We have a lot of entomologists. We do a lot with high throughput phenotyping, cropping systems. Again, every gene only makes sense in context. You make a loaf of bread, all wheat has to be looked at for quality. We have our own quality laboratories. Wheat is basically grown on ground that's too poor for corn or soybeans. So our uniformity completely lacks compared to what the best corn ground is. You know, the worst corn ground would be the best wheat ground is what it comes down to. So we have a lot of spatial issues when it comes to doing evaluations. We work with statistics and biometry. We do some crop modeling. I also work on triticale. I work on barley. I think they're great crops. Really good technical people. We never are rich, but we always nickel and dime our way to middle class, so that's our funding sources. With that, I'll say thank you and ask if you have any questions.
Yeah, we have some time for some questions, so please go ahead. And if just a reminder, too, if you're going to ask questions, please use the microphone. Thank you.
Thanks for a nice talk. I think that we typically view the role of landraces in a breeding program as conferring disease resistance. At least that's the context in which we usually talk about them. But I'm curious if there's any instances in wheat where we've used landraces to explore flavor or quality traits that you know of.
Oh, it's an excellent question. For flavor or quality traits, not so much in wheat, but you certainly have done that in tomatoes. You know, I mean, again, in wheat, we have, I'm not sure we actually have what I would call flavor components that are well characterized. If we did, I think we would. We have used landraces, we have used wild wheats to expand the glutanins and gliadins, okay, because there's a great diversity in those gene families. And so we have looked at those, okay? But that's mainly for functionality. It's not so much for flavor, okay? But in species where flavor is important, I am sure that they have used the wild relatives and their equivalent of landraces to do that, absolutely. We also use landraces to get inside the ballpark, you might say, of what the uses are. I mean, quite frankly, a leavened bread is completely different than a European bread, which is actually whipped into its shape. You might say it's a high-speed mixer. They beat air into it. They don't use yeast, okay? That's different than a flat bread, which is used in the Middle East. And so if you're looking at characteristics, you'll often go for those types of things to find out what the indigenous race is used for that. Absolutely. Okay?
Could you please comment on how war in the Middle East is affecting germplasm exchange and storage?
Oh, okay, okay. So, question on war in the Middle East, or we'll just call it turmoil in the Middle East, okay? And germplasm exchange and storage. The storage is always a problem, okay? And Arnold can probably talk to you a lot better than I can about what turmoil in Central America did to the great corn collections where they were indigenous. And so, things like this. When Afghanistan went through its, well, I'm not sure if it's finished yet, but it's basically 30 years of war. After, our American efforts there, the first thing they did is they went back to the collection and they re-brought the Afghan wheats back to Afghanistan because their agriculture was decimated, and places like Icarta and Simit had stored representative germplasm from Afghanistan. So they immediately brought back from their storage collections, which were in secure sites, the wheat back into Afghanistan. Okay. Now, I got to tell you a funny story about turmoil in the Middle East, and I'm not sure this is quite kosher, but a very good friend of mine is a man by the name of Moshe Feldman. He was a great cytogeneticist and wheat evolutionist, okay? And he is in Israel, and everybody from the age of, I think, 18 to 55 serves in the military, okay? And Moshe was past 55, but his son was serving. So Moshe's son, was on mission. He came home, and Moshe always shined his shoes so he'd look good. So his son wouldn't have to do that. And so he was shining his shoes, and he noticed there was a seed in the shoelace. Now Moshe knows every wheat in Israel, and he said, "That's not from Israel." So the next morning, his son wakes up and he says, "Son, where were you?" "Oh, we were on a mission." He said, "Yeah, but where was that mission?" "Oh," he said, "It was up in northern Israel." Moshe said, "I don't believe you." Okay. His son kind of looked at him and he says, "So where were you really?" "Well, they had gone into Syria." And he picked up a seed and come back. So that may be a beneficial aspect of germplasm exchange. Okay? We don't recommend it. Okay? We don't recommend it. But you will see germplasm exchange is unique. There's a wheat that was a very important stem rust gene called Kenya Farmer. And back in the day, England and, at the time, Tanganyika, which was under German control before First World War, didn't share germplasm, okay? And so a Kenya farmer walked in and he saw a stem rust-resistant line, and he took the seed and he put it in his sock, folded it over, and walked out of Tanganyika with the seed. So germplasm exchange has a way of working. If you have a friend, you usually can get a lot of things. You never divulge your sources, but it's good to have a lot of friends. Okay, that's what I would say. The storage is the critical part. And there's two types of storage. One is the in vitro collections, okay, and the second part is the in vivo collections. And most people have living collections in the areas where it's indigenous. And just to give you an example, and again, if you're lucky, I remember being at the Sea of Galilee, looking at a boulder that had like a pocket in it with maybe a handful of soil, and a Hordeum spontaneum had landed in there, and this is a pretty desert country, had enough moisture that it went to flower out of this boulder in this cup of soil. So you can see some amazing things. If you really want drought tolerance, if you go to the Negev desert, the roads, will slope, and there will be a slight puddle for when it gets its 100th inch of rain, and you'll see Aegilops longissima growing where that little ditch is. Everywhere else, there's not a plant in sight, but where the moisture ran down and it puddled up for just a brief moment, you'll see Aegilops longissima. It's absolutely unbelievable.
Okay. Thanks for the nice talk. I'm Xiao Chen. So I have a question about the ABD genome. When you do these combinations, you're looking for a high yield, right? Mm-hmm. So I'm studying stress response. So I'm interested in whether these different genomes are correlated to different stress conditions?
It's a great question. So for those who didn't hear it, are the genomes correlated to different stress conditions? Well, one of the things we could show is that most people believe that a lot of the winter hardiness came in from the D genome, okay? So that's a stress that it would be involved with. What we would say is the stresses would depend upon where the species came from, okay? So if you went, like I said, to the Negev desert, which is extremely dry, and you looked for Aegilops longissima, which is the predominant wild relative of wheat that would be found in the Negev, you would certainly have a correlation with that genome and stress, okay? As you go to northern Israel, you'll see things like the Uartas and whatnot. There's a lot of different things. It's a much wetter climate there. You know, the cedars of Lebanon Lebanon's actually a relatively wet climate for the Middle East. So you will see that. But it really comes back to if the wild species and either the abiotic stress or the biotic stress are endemic in the same area, or the landrace, you'll find the traits you're usually looking for. I mean, nature's a great selector. And again, what you have to remember is like I said, the seeding rate, if you've ever, and again, you probably haven't, but you will see a hillside of Hordeum spontaneum, which is the wild relative of cultivated barley, and it will look better than a commercial stand of barley. I mean, it's that thick, okay? So the chance for selection, chance for mutation is as high in that as there is in any commercial field. And you've got all this heterogeneity, you've got a low level of outcrossing occurring. So it's quite remarkable what you can find. But where you would go is, I mean, so like, you know, to give you an example, the center of origin of wheat is in the Fertile Crescent, okay? The secondary center, and where you would go if you wanted barley yellow dwarf virus resistance, is Ethiopia, okay? So you have primary centers, then you can have secondary centers where things moved and then they evolved, and what was endemic there became important in the structure of those populations. So, you know, you may not think, like, I wouldn't go to northern Israel to get drought tolerance, okay? Now, you may find it there because it's random chance, okay? It can be random chance. But the highest likelihood would be where the drought would be, which would be in their moisture gradient as you go to the Negev. And it would, the same thing would be as if you're getting out of the irrigated cultures of the Tigris and Euphrates to the dry lands of Iraq, those areas, and in Syria. Did that answer your question? Okay.
And if there are no more questions, let's thank our speakers one more time.
Thank you.